denaturing. And that is where you break the bonds, the hydrogen bonds of the proteins. And that means they're no longer going to have the shape to work. So if you get a fever of 106, maybe even a little bit less than that, you start to have problems holding together all those enzymes that are in your body, and your brain is the most active organism in your body. And so it's going to have a lot of enzymes doing a lot of stuff, and you're going to start breaking those things down, and your nerve cells in your brain are going to die. Okay? And your brain will stop working, and your brain will stop telling the rest of the body what to do, and then you're dead. Okay? So we'll go into why fevers are, in fact, as I'll go into it now, a small fever. Now, when you have a slight temperature, like 101, 101, you have that temperature because your immune system is trying to slow down the invader, be it a bacteria, a virus, a small single-celled eukaryotic organism like some amoeba and things. Your immune system, the generalized immune system, has found the invader, but it needs to make antibodies, which are specific warriors that go specifically after that invader, whatever it is. Well, that's going to take about 2, 3, 4 days between when your immune system first recognizes that it's being invaded and the time you've built up all your antibodies. Okay? And we'll talk about antibodies more in a genetics lab. Okay? But for right now, antibodies are the army that's going to, very specific army to go after a very specific invader. Okay? And during that time, between when you first get it detected, and now while your army of antibodies is getting made, you need to slow down the invader to keep it from multiplying. And to multiply from, say, 1 virus to 2 or 20 or 1 million, they need more proteins. They do, the invaders, to make new proteins. So if you heat up your body a little bit, it's harder to make proteins. Okay? You may not at 100 degrees or 101 break down any proteins, but when things are starting to move a little faster, it's harder to make these bonds right here, the secondary structures. It's harder to make them when things are moving quicker than they're supposed to. And so when you get a fever, it's got to function, and that's to slow down the invaders while your immune system makes an army to attack the invaders. Okay? Does that make sense? So let me ask you this. When you do have a fever, what happens to your appetite? Are you hungry or not? And the reason is to eat food, you need to make digestive enzymes. And the body says, forget it. You can't make digestive enzymes right now because you got a fever. And the reason you got a fever is because you're trying to fight an invader. All right? So it automatically cuts down your desire to eat, because trying to make digestive enzymes, you might be able to make some, but you're not going to make them all. So you're going to be eating food that you cannot digest, which may enhance your illness. So in that case, we just don't do it. We just stop trying to eat, so that when we try to make that digestive enzyme to eat our food, we can't, because it's a little too warm. Now, most enzymes will hold together up until about 104, 105, 106. And so any enzymes you did make at around 104, 105, 106, there's variation in that. The enzyme can start, in the protein, start to break down. This is why having a small fever is okay, because that's how your immune system is supposed to work. But getting up too high is dangerous because you can start to denature enzymes that are extremely important for life. Okay? Does that all make sense? So when you get a fever, as long as it's somewhat low, don't do any. I'm not a doctor, but I think most doctors are going to tell you, okay, you got a fever, that's good. It's doing what it's supposed to do. It's slowing down the invaders until your immune system gets going. Okay? Until your immune system is up to speed with very, very specific antibodies to attack that particular invader. This is why if you got the vaccines for COVID, usually the next day you might have had a fever, okay? Because they injected into you, depending on which one you had, but most of them were making what we call the antigens, which are the markers of the outside of the virus, and your immune system finds them and goes, ooh, we're being invaded. Let's make antibodies. But it's going to take a few days to make the antibodies. But that first day, usually you get a fever, because a slight fever, because that's what your immune system does when it thinks it's being invaded. Okay? So that's a normal immune response to a vaccine, is you normally get if it's working, you normally get a little fever. Okay? And it may be very short-lived. Okay? So anyway, denaturing. So we're going to look today at how denaturing, the breakdown of the normal structure of a protein, okay, we're going to look at how that functions and what does denature a protein, okay? And if it denatures, that means it doesn't work, okay? And what enzymes do is they take a substrate and they turn it into a product, okay? That's what they do. And again, you have tens of thousands of enzymes in you. Some of them are for breaking things down from big polymers down to monomer form. Other enzymes are there to take monomers to make them into polymers for carbohydrates, for lipids, for proteins, that kind of stuff. Okay? So that's what we're doing. So we're going to take a disaccharide, okay, and that disaccharide, that's two monomers of sugar. Saccharides are the Lego blocks of carbohydrates. And so when we have two Lego blocks, two monomers together, it's a disaccharide. And we're going to take those two and break them down with the enzyme that we are going to use to their monomer form, monosaccharides. And in this case, sucrose. Sucrose is the disaccharide. Come on, let me see where I'm at. There it is. Glucose and fructose are the products, and they are monosaccharides. So when we look at, here's sucrose right there. I'm going to make this a different color so I can see it easier. So here's sucrose. Two monosaccharides stuck together. And we have to do hydrolysis to break them down. And that's why there's water here. So it's sucrose plus water. Sucrose plus water is going to give us glucose plus fructose. So this is what it looks like chemically. Okay, so we have the sucrose right here, okay, plus the water. And this invertase is the tool that's going to add that water in a very particular place. And when it adds the water, boom, we end up separating the glucose from the fructose. Okay? That's what we're going to do today. That's all today is about, is looking at how this works in different settings. Everybody understand that?
I have a question.
Sure.
Okay, so I just want to make sure. So the products are the sucrose and H2O. And
No, stop. The products, okay, you own a company, it's what you put out is the product.
Oh, okay.
I mean, think about it. You've got a company, your product is what you sell to people. It's what you put out. So what is the substrate?
That would be the sucrose and H2O?
Yes.
And then the product would be the glucose and fructose. And the invertase works as a catalyst to speed up the process, essentially.
Yeah, enzymes are the tools. You want to build a house with 2x4 or with wood. You got the wood, you got the nail. Okay, you want to get them together, and if you're a martial arts expert, if you want to ruin your hand over time, you can try to hammer them in with your hand. Okay, but most people use a tool we call a hammer. Okay, to drive the nail into the wood. The invertase is the tool that's going to put the water in the sucrose, and that will split that disaccharide into two monosaccharides. Okay. So that tells us right there what is the substrate. You guys are supposed to answer what is the substrate and what are the products. And you write those down here. Okay. And the answer for that is right up here. Remember, products are what's being produced. Okay. Something that's being produced to be sold. In this case, you're selling this stuff to our digestive, or the digestive system's putting in our bloodstream, and now it's part of the stuff in the soup of the bloodstream that's going around saying, "Hey, I'm for sale. You want me? Grab me. Okay, take me in." Okay. If a student mixes some sucrose, which is the substrate, with invertase, which is the tool, the enzyme, and a test tube allows some for reaction, what results would you predict? Okay, so you need to come up with an answer for that. Does anybody know what the answer should be for question 2? What's the answer for question 2?
My guess would that the sucrose to break down into fructose and glucose?
Yeah, so that's your results would be the sucrose would break down because of hydrolysis into glucose and fructose. That's it. So that's what we're doing today. But we're also looking at what is going to be impacting the process. So the problem, let me show you before I get to the Benedict solution. Okay, so here's sucrose, there's fructose, and there's glucose, and there's water. If you just took the labels off and looked at them, they all look like water. So if you took sucrose and you put invertase with it, they should, according to what we know, turn into fructose and glucose. How could you know that? Because you're looking at it and everything looks like water. Okay. I mean, if you had a very discerning taste buds, you might be able to taste the difference between sucrose and fructose and glucose. I can't, but if I know that I buy cheap wine because I can't tell the difference between $80 bottle of wine or a $10 bottle of wine. So why buy the $80 bottle wine since I can't taste the difference? But you might be able to taste the difference, but it's easier if you can see it. And that's the role of the Benedict's test. Okay? The Benedict's test, I'm going to make this bigger so I can see it again. Okay, the Benedict's test is all about detecting, okay, if something happened. Okay. So Benedict's is a blue color. Okay. And what's the definition of a compound? What's the basic definition of a compound? I don't want to have to ask this for points, but you guys really need to know. 2 or more elements in a fixed ratio. You got to have that fixed ratio. Okay. 2 or more elements in a fixed ratio. And we see that with water and hydrogen peroxide. Both of them are just hydrogen and oxygen, but the ratio is different. And so it gives it a different chemical reaction. Does everybody understand that? Because of the difference in the ratios, we get a different chemical reaction. And you really have to understand that. And I want you to accept that. So please, if you don't understand it, let me know. Because
I'm sorry, but just to make sure, so if you were to combine these 2 different solutions, you would get a new reaction?
Any compound, if you add or take away something, if you add to that compound or you take away from that compound, you're going to change its chemical reaction it's going to do.
Okay.
What it looks like. Does that make sense?
Yes, that makes sense.
Okay. So Benedict's solution is a compound. Okay. But it will react with monosaccharides. When it comes in the presence of monosaccharides, it binds to them. So they become one. So what was Benedict's solution is now Benedict's solution with a sucrose. And it's now something different. Does that make sense? So a compound is 2 or more elements in a fixed ratio. And every compound has its own chemical properties. And those chemical properties include possibly color. And this is how astrophysicists look at stars or distant objects and can sort of get an idea about the chemical makeup is because of what light is coming out of it. What are the different types of light coming from that star or other body in space is because the different chemicals give off different light. Okay. And we talk about Benedict's solution, it gives off blue. We see blue with Benedict's. However, when it binds with a sucrose, with a monosaccharide, like glucose or like fructose, then it changes and it puts out a brownish color instead. Okay, so that way we can use the Benedict's test to see if there is any glucose or fructose in a solution, okay, by doing this Benedict's test. Because if it's sucrose, if fructose or glucose is present as monosaccharides, they bind with Benedict's and make it turn brown. Okay, it was blue and now it's brown. And we go, oh, the Benedict's found some monosaccharides. So we're going to use this test to see if, in fact, we can take sucrose, which is a disaccharide that does not react with Benedict's, and put it with an enzyme that will take sucrose, a disaccharide, and turn it into two monosaccharides, and we can go with B. We just found, some monosaccharides that weren't there before. They weren't there before, but now they are. So it must have been the enzyme invertase that made that happen. Okay. And so when we look at the results for how much, it's how deep was the change in color. If there's no change in color at all, you put negative. So if blue, the Benedict stays blue, that means there was no chemical reaction. If it's got a little bit of brown to it, a hint of brown, you put 1+. If there's a little bit, and this is subjective, if you think there's more, if you think, put 2. If you think there's a lot of brown, you put 3. When I say brown, it looks like muddy water. You know what, water, and you get mud in it, it looks brown. And so when we look at how much mud is in the water, okay, how much mud is there, that is the Benedict's test. Okay. So we're going to look at this Benedict's test, and the way to do it, and this is the way it's done all the way through the 3 different labs that we're doing today, is the test solution is the stuff you think something should have happened. Okay, you put the invertase with the sucrose, and you give it some time, and you go, okay, I'm going to see if it worked, and that's the stuff you're checking to see if it worked. The Benedict's is going to indicate to you if it worked or not. So you got 2 mLs of what we call the test solution, which is what you're checking, and 2 mLs of the Benedict's. And then what you're going to do is you put them in boiling water, because the Benedict's test doesn't do well unless everything is moving quickly. So you put them in boiling water for a few minutes, and then you take it out and look at it. Okay. So we're going to be looking, and these are the steps. And we're not going to go too crazy having to memorize this, but there's certain sequences. And if we're in the lab, your group, you'd have 3 to 4 people in a group. One person is going to be checking the blocks. So you do step 2. Well, obviously you turn on the hot plate, but as you do these steps, you check them off to make sure that you're doing all the steps. Okay? And you do all the steps exactly as they say. Okay? And then you do this, that's supposed to be 6. What? Somebody screw up there. Yeah, that's supposed to be a 6. Those are 5, 1, 6 or 5, 1, 7. That's supposed to be a 6. Okay. But you're doing these steps one at a time in sequence. Okay. And then when you're done, you're going to interpret the test. What we're doing today first is we're taking sucrose, 1% sucrose. How much water is in the sucrose? If this is a sugar solution, yeah, you got 99% water 99% water, 99% water, and 100% water. This is 100% water. Okay? So we have 4 test tubes. This is a disaccharide. This is a monosaccharide. And this is a monosaccharide. And remember, Benedict's test, the Benedict's solution, that compound that we call the Benedict solution, that reagent, which is over here, which is blue, if it comes in contact with either of these, it turns brown. Okay, so we want to see what happens. And you're going to look at this, and you go, okay, we have 4 test tubes. Okay, and we come back down over to here. Test tube 1 is sucrose. Sucrose is what? A disaccharide or a monosaccharide? What is sucrose? What is sucrose? A monosaccharide or a disaccharide? Yeah. Does Benedict's react with a disaccharide? And so in test tube 1, that's what's there. Is there a reaction? And the answer is no. We look at test tubes 2 and 3. One of them, test tube 2, has fructose, which is a monosaccharide. Okay. Test tube 3 is glucose, which is a monosaccharide. Both of them turn a color. And we have tables today. Every one of these tables is pretty much alike, except for what's in the test solution, or what the test is. You're going to write down what color you see in the tubes. Okay? So you're going to put down in test tube 1, it looks blue. Test tube 2 looks brown. Test tube 3 looks brown. Test tube 4 looks blue. Okay? And you're going to write that down. Now, for the Benedict's results, did the Benedict's react to what was in test tube 1? Yes or no? How would you know?
No.
And how do we know that?
It stayed blue.
Exactly. What about this one? Did it react?
Yes.
Test tube 2. It reacted, and it reacted pretty good, so, might as well put 3 little things there. Okay. 3 little plus marks saying it reacted pretty well. And then the glucose, 1% glucose, did it react? Yes or no?
Yes.
Yeah, because what color is it?
Brown.
What color is test tube 3?
It's brown.
That means it found it. Benedict solution found a monosaccharide to bind with, and it did. Okay, what about DI water? Did it react?
No.
No, because what's in water? Is there any monosaccharides in pure water?
No.
No.
Obviously not. So remember, you guys, you have to write down what colors you see that are right there. Okay. Now you got to answer some questions. 4A and 4B. Okay. And this is about, you're looking at somebody that works in a lab. They're there on a Saturday, and they want to find out if the invertase is any good. Does it still work? Does it still work or not? And so they tell you about what she does. Okay. And then you're going to have to answer questions A and questions B based on what you found out in the information from question there. Okay. Now, the first real activity we're going to do today is the effect of temperature on invertase activity. So the very first thing you have to know is when you stick a thermometer into something, what are you checking?
Is it temperature?
What does the temperature indicate?
How fast something's going.
Exactly. How fast or how slow? And so we look at an enzyme, okay, and I'll draw it this way. So we look at an enzyme. This right here is called the active site. So I'm going to do this. So we have substrate over here, and we're going to have product over here. And what happens is the substrate goes into the active site. And in this case, water is also going to go into that active site to make, in the end, a product. So what is the substrate in this experiment? In this whole today, there's lots of possibilities, but in today's, what are we using? We're using sucrose. That is the substrate. What are the products? What are we making? Right here. What's being made? Fructose and glucose. Okay? And they are reducing sugars, so they should react with the Benedict's test when we're finished. But first we have to do this. We have to do this. Okay? So what is the name of this enzyme? What's the name of that enzyme? Anybody remember the name of the enzyme? The tool. Yep, it's invertase. Anytime you see this ASE, that's going to be usually an enzyme. Okay. Alcohol dehydrogenase. Okay. ASE, that's an enzyme that helps break down alcohol in your bloodstream. Okay. ASE, ASE. So there's all kinds of enzymes, and you can usually, not all, but usually when it says ASE, that's an enzyme. But not all enzymes, and with ASE, there's a few out there that aren't. So this is an enzyme. This is the tool. We got the substrate, which is sucrose. We got the product, which is glucose and fructose. Can you guys see that there's movement that's going to have to happen? Does not the substrate need to move to the active site? Because if it doesn't move to the active site, how is it you're going to make a product? Does that make sense? The substrate has to move. And it's going to be in a solution. In real life, it's in your cells, okay, or in your digestive system. But it's in you, okay? In this experiment, we're going to have it in different environments of temperature. And temperature is about the speed of motion. So if we have something that's really cold, okay, if it's really cold, that means it's moving really slow, okay? That's moving really slow. So that means production is going to be really slow. You owned a factory, and there was some person that had to get this this substrate over to the enzyme, but they would, walk 5 feet, talk, and take a sip of their coffee, talk to somebody about, what would happen this weekend or what's going to happen. Take another 5 steps, run to somebody else, and you're the boss, and you're going, how come our production is so slow? And you see Gary, who's the guy who's taking the substrate to the machine, to the tool to make a product, is moving really slowly. And you're going, okay, I'm going to have to put a fire under Gary's butt to get him to move faster, because we need to make more product, because we got to make some money here. Okay. So cold is slow. And then we get, a different, room temperature. Okay. It's going to move a little faster. Okay. That's going to be a little faster. Okay. At room temperature. And then we have actually we reduced it down to 39. Oops. Sorry. I think we reduced it down to 39. This is Celsius, and this was at 20 degrees Celsius. Okay, and this is going to be faster. Okay. And we also have 100 degrees Celsius, which is really fastest. So what we'd expect to see is the faster we make things move, the faster we're going to turn out a product. Does that all make sense?
Yes.
All right. But what happens when you get too hot in your body to the enzyme's structure? It denatures. So when you take that at 100 degrees Celsius, these are Celsius, this is boiling water. When you get that that denatures the enzyme, the tool. And if you denature the tool, then you could move the substrate as fast as you want, and it's still not going to turn into a product, because you basically broke your tool. So when Gary is moving quickly and brings it over to the tool, but the tool is broken, you're still not going to make a product. And the manager, the owner of the company, is going to be mad because you're not making a product. But it's not Gary's fault anymore. It's because you broke the tool. Okay. So that's what we're doing with today's experiment. Okay. Now, when you're looking at these terms, okay, and I'm trying to get that to where I can see both. We have these dependent variable and independent variable. Okay. And remember, when you're looking at a graph, you got something down here, okay, and you got something over here. The something over here is usually what we call the dependent variable. And the reason there's a line is it varies. So a variable varies. And here we got something else. Okay. Okay. Here we have something else. That's the variable. But one of these is the dependent variable, and one of them is independent. Okay. And what we're looking at today is the effect of temperature. Do the temperatures vary?
Yes.
Okay. And if we go back to my little thing, is temperature going to have an impact on product?
Yes.
So which one? Both of them are going to vary, both temperature and production. Which one is dependent and which one's independent? Okay. And that you need to write down. Okay. And then a hypothesis is an educated guess. When you expose,
Professor, I have a question. So you're saying that one of the either the temperature is the dependent or independent, and what's the other thing that we're using? I don't understand.
see my circling?
Mm-hmm.
What am I circling?
The product.
You got it. Production.
Okay.
You know, in a factory, you can measure production by how fast or slow Gary is walking. Once the raw material, the substrate, gets to the tool, it makes a product almost instantaneously. But it's got to get there. And if Gary is walking slowly from the store where they store the substrate and walk slow, you're going to make slow products. I mean, you're going to make a product slowly. If Gary's walking faster, it's going to be more production. Okay. So what's dependent on Gary walking? How much production we have. Okay.
Temperature?
So temperature, the speed of motion. Temperature is speed. So the faster you're going, the more products you should make. Okay. So your hypothesis is an educated guess of what you expect to happen. To answer your question, how does temperature have an impact on production? How does temperature impact production? And you put down if the temperature goes in a particular direction, and then I put my substrate and enzyme together, then I should expect this kind of production.
So we're not actually testing for the invertase activity. We're testing for the amount of products made.
How do you know if invertase activity worked? If this is
color.
Okay, but let's go this way. So this is a factory, and I'm taking raw plastics. I work in this factory, and I'm taking the raw plastics to a place right here, to this active site, and I put that raw plastic in the machine, and it takes and makes, a plastic doll. Okay, it makes a plastic doll. So when I look at my enzyme activity, I can say that this invertase activity is high if my doll production went up. The only way I can measure the enzyme activity is by measuring the product. Is it more or is it less? Or is it non-existent? Does that make sense?
Yeah, so I guess what I'm saying is when the substrate is brought to the active site, the invertase is going to act the same speed regardless, right? So
Yes.
Gary, in this example, is actually the temperature because
Yes.
we're... Okay.
Exactly. How fast I'm walking.
Okay, so the actual reaction with the invertase never changes rate of speed.
No. But
Okay.
it can only put out. I mean, if you hire someone to make a plastic doll, and it all depends on keeping the invertase, this mold thing, fed with raw plastic, then how fast this turns out the doll depends on how fast you feed it.
Got it.
Because if it's being fed slow, it's going to make product slowly. If you feed it quickly, then you have quicker product production. Does that make sense?
Yeah, thank you.
Sure. Yeah, so when you're looking at the dependent variable, it is the enzyme activity. And the independent variable, which varies from 0 to 100, is the temperature. So the temperatures are going to go down here, and the independent or the dependent, which is production, is going to go there. Okay, and you state your hypothesis, and then we do the experiment. Okay, and you would follow these cookbook exactly. Again, someone in your group would be the check person. Okay, we got this. Next, we got that. Next, we got that. You don't just haphazardly do it. You go through this checklist, okay, and what you have is you're going to have tubes, 4 tubes, and you're going to put down, the enzyme in them, a solution, okay? You're going to put them in here. So they're all going to have the same amount of enzyme, okay? All of them. But you're going to label one of them for 0. One of them is going to be 25. One's going to be 37. And one of these is going to be 100. So you mark those for the temperature, and you do that with little tape. Put a little tape on there and mark down which temperature. And then you're going to put 2 mLs of the enzyme. And what is the enzyme? What is the enzyme today? Invertase. So you're going to put the invertase solution and 2 mLs of buffer. And I should talk about buffer right now. Okay. Buffers are substances that, when in a solution, will try to keep the pH where it is. So you can design buffers, which chemists do, to say, I'm going to keep this at 4.4, a pH of 4.4. And no matter what kind of chemical reaction occurs in there, that pH will stay the same. Because the problem is, when you do chemical reactions, the products often change the pH because they're going to give off possibly hydrogen ions or hydroxyl ions, and that's going to change the pH. And we're not interested in that today, okay? We're not interested in this experiment about the changing in pH, okay? We're interested in temperature. So a buffer is a chemical substance that is going to resist the change in pH that's caused by chemical activity. Does that make sense? So I'm going to write it over here. So buffers are chemical agents that will allow a solution to maintain a specific pH in the face of chemical reactions. Normally would change the pH. So does everybody understand that? That's why they're called buffers. You take buffered aspirin, some people do, because aspirin by itself, in some people, when it hits the stomach, there's a chemical reaction that causes the stomach to become even more acidic. And people who are prone to ulcers, taking raw aspirin can eat a hole right through their stomach because it changes the pH. So they take buffered aspirin. So during its journey through the stomach, the buffer that's included in the aspirin will keep the pH of the stomach the same because we don't want it to change. Okay. So we're going to include a buffer solution so that we don't want any chemistry. So step 1B here is to do that to all the tubes. Okay, and then we tap it a little bit to make sure it's all mixed up. And then we have 4 tubes, and those 4 tubes are going to have the substrate in them. Today, what is the substrate of those tubes? What is the substrate of the tubes? It's good, sucrose. And then we let them sit in there, and you're going to do this. You place all 8 tubes, the ones that are marked for 0, in the ice bath, or you have 8 tubes, so it's going to be a substrate plus an enzyme tube, 2 different tubes in the 0. The same thing goes for 25, 37, and 100, and you let them sit there, for 10 minutes. And then you mix them together, okay, which is what it says to do right there. You mix them together, and you put them back in to the solution, the mixed part of it, for another 10 minutes. And then you do a Benedict's test, okay? So you're going to take some 2 mLs of your Benedict's and 2 mLs of your stuff, and you're going to put them in boiling water. Make sure you keep track of what's what, and you take them out, and this is what they look like. Okay. So this is test tube 1, 2, 3, and 4. And you're going to write down, what color they are, and if any precipitate, any mud was performed or showed up in these tubes. Okay. So we're going to take a break, okay, and we're going to come back at 12:35, okay, 12:35 on computer time, not your clock on the wall. Okay. So make sure you look at that and understand it, what you're going to be writing down for these tubes that are going to be down here, and what that means. What does that color mean? Okay. I'm going to stop my share. I'm going to pause the recording.
Recording stopped.
And then we're going to take a little break, okay? Till 12:35.